present here COVID-19 disease map, a systems biology resource to study molecular mechanisms of virus-host interactions. We initiated this project back in February 2020, in the beginning of the pandemic breakout. The ultimate goal is to find a treatment and to develop a vaccine that would stop expansion of the infection. To reach this goal, we need to understand the molecular mechanisms behind this disease. The entry point was the disease map community, mastering various tools and applications to study molecular mechanisms of different human diseases. Number of teams in the community initiated the COVID-19 disease map project. The project aims to gather together the molecular information and then using this map to analyze the data from the disease and to suggest intervention points and drug repositioning. The project is open to the entire world, and indeed several teams from different countries joined us. The project has two major pillars. The first is knowledge formalization, namely representation of information from the literature in the form of modular disease map. Then the map applications. For example, multi-omics and clinical data analysis from COVID-19 cohorts, and then drug repositioning, modeling response to treatment, and also envisaging disease comorbidity. The project is multi-step and multi-layer. We are numerous teams and also mastering different expertises. Therefore, we divided tasks. The first one is creation of the map, gathering the knowledge into the formalized representation of the entire COVID-19 mechanisms. This is the task of curators. Then the resulting maps are checked by the domain experts. The curators retrieve information from the literature and represent this information in the form of process description diagrams using systems biology standards and using official identifiers for naming the entities on the maps. The processes implicated into COVID-19 disease are numerous and various. Therefore, to divide the work, we had to imagine the mental map of the disease. Here in this scheme, we represent the interaction between the virus and the host cell and the processes that take place inside the host cell. Then there is a systemic response, mainly the immune response, and further the repair and recovery mechanisms. The curation, manual curation, is time consuming. Therefore, we decided to use also the text mining techniques. Several teams that joined the effort suggested various flavors of approaches. Using these text mining approaches, we could retrieve a body of literature which is specific to COVID-19 mechanisms. And these articles were used by curators to enrich the initial maps. Of course, this collection of the maps need to be gathered together under one roof. And this is MINERVA platform, a web tool that allows a permanent update of the maps and also exploration of the maps and different zoom levels and data analysis in the context of maps using the toolbox. These carefully curated and annotated maps that are gathered under one platform are now ready for other applications, namely for data analysis and for modeling intervention points and drug repositioning. The initial maps are quite complex and comprehensive. These are process description diagrams that are suitable for data integration. However, to perform modeling, one needs to go to another level of representations, namely activity flow diagrams. There is a converter from process description to activity flow diagrams integrated into the workflow. These activity flow diagrams then can be directly used for mathematical modeling. And indeed, the preliminary results of the modeling already suggested at least one intervention point. And this point is druggable, and there is a drug that exists in the drug repository, and this is an approved molecule. Therefore, here we also suggest a drug repositioning. Of course, this is an ongoing project. We suggest here a disease map repository and also a computational workflow. The entire framework can be applied for studying other human diseases. We keep the resource updated thanks to automated text mining pipelines and manual curation. The analytical workflow is adaptive and flexible, therefore, evolving tools can be integrated. The web platform is easily accessible to computational and non-computational users. This is a community effort. Therefore, thanks to interdisciplinary teams, we will finally suggest a thoughtful drug repositioning that can be reached out to the companies or other teams and justify the choice of drug development. This project is an international initiative. There are multiple contributions, a wide range of expertises and domains, and the repository is growing. The entire content, documentation, and code are available.